Good afternoon. Thank you for being with us today on site and online. It's always a pleasure to meet people in person. I'm delighted to welcome today Dr. Wouter Saelens. Wouter is a scientist in the Deplancke lab, the Laboratory of Systems Biology and Genetics here at EPFL. His talk today is titled Mountains in Our DNA, How Combining Probabilistic Modeling with Neural Networks Deepens Our Understanding of Omics Data and Genetic Variation. You know now the concept. We have roughly 1 hour together. I think Wouter planned for 45 to 50 minutes for his presentation. He kindly agreed, if you have any urgent questions, to signal them directly, either in chat or by raising your hand, or to keep them until the Q&A at the end. Before we dive into the presentation, I would like to thank Wouter once again for his availability and the entire CRS team for having made this event possible today again. Now, Wouter, floor is yours.
Thank you. All right, so I'm going to talk about mountains in our DNA. Didn't know those existed, but they do exist, at least according to us. So what we are interested in the Deplancke lab is genetic variation. Yeah, because as you probably all know, genetic variation is a major driver of a lot of diseases. So here's just a couple of examples where often it's about 50% that is driven by some genetics, as far as we know. Yeah, using some twin studies, for example, that's been studied in this paper. Sometimes it's a bit more, sometimes it's a bit less. There are, of course, a lot of other factors as well, but we are really interested in the genetic cause because that is still not really well studied yet, not well known yet. And in particular, like what do we actually want to do? We want to do precision medicine in the long run. And essentially, precision medicine is trying to use some data from the patient, for example, the genotype, to specify which drug, which screen we have to do to understand whether the patient has a disease or will have a disease, whether it will relapse or not. Now, the problem is this link is super difficult to do because there are a lot of layers in between. Between the genotype, a lot needs to happen until you have a disease or until you are more susceptible to a drug. And the problem is it's very complex and we have little data. And the data we have is really hard to interpret. That's why in the Deplancke lab, we try to focus on the intermediate layers. By mapping these intermediate layers, hopefully we will be able to have a better predictive model. And the layers we focus on are like genotype, the chromatin state, the transcriptome, and the overall cell state, which all influence each other and which will hopefully be contain some predictive information. So this is our focus. And I'll start with kind of discussing this chromatin state. So that's the state of our DNA and how it is actually contained within our cells. So our DNA, as you can see here, it's very long. And the problem is then that it's present in every cell and needs to, in some way, be kind of put together and condensed in a certain molecular state, and that is called the chromatin. And the chromatin, so we have it in all of our cells, and we really like to study it because the chromatin kind of tells us then which parts of the DNA are active and which parts are not active. And as you can see, some parts of the DNA are wrapped around these things called nucleosomes, but other parts of the DNA are open. They are accessible. And this is really crucial because DNA that is accessible, it can be seen by other proteins, by other molecules in our cell. And this is then, being able to see this DNA is then able to also use this information to, for example, regulate then gene expression. And so we study this open chromatin, this DNA that is accessible. Of course, we cannot really directly see it through a microscope, but what we can do is we can kind of use some techniques called ataxic that you get this type of data, which is essentially fragments where the DNA was seemingly open at the time inside of a particular cell in our body or from a patient. And so how the data looks like is we have a start and an end, and we know in which cell this DNA was open. And now, so this is the raw data we're working with. It doesn't really look like a lot of data that other people work with. Maybe looks a little bit like spectroscopy data, perhaps. It's one-dimensional, and we want to now use this data to learn things about the cell state, about the genotype, and so on. So I'm now going to show you how the data then actually looks like that we get from a particular gene. It's the IL-1B gene. We're looking around this gene, and here you see the individual fragments. Where is this gene open? In this cell. So each row here is a single cell. And the cells are ordered by how much the gene is actually expressed. So here the gene is not expressed, but still the gene is a little bit open. And you can see it's very noisy data, but still there's some structure there, right? So certain parts are open in this case. And then we can go up, we can look for, so the gene is still not expressed, but at some point the gene is expressed. And then you see that the data is changing. So the DNA is opening more at particular regions, not everywhere. So maybe here it's not really opening much more, but here it's clearly opening, and also here in the gene body.
Just to clarify, so here you're saying that in some subset of cells, there's more availability to the DNA.
Yes.
Okay. By saying it's opening more, you mean these cells, it's more open.
Exactly.
Okay.
Yeah.
Is there time here or no?
No. Yeah. So these are actually blood cells. So kind of these cells are already mature in our blood. But we are looking also at time data sets. Super interesting. Yeah.
Can you say with certainty that in the white areas, those genes will never be expressed? Like it's really a gate. You have to have the open state and then the transcription factors associated with the gene. Or can you say that if there's a closed state, it still might be expressed?
Oh, it super depends. Yeah, you have some genes that are closed, still expressed. Other genes that are open, not really expressed. So that's exactly what we try to understand as well. Overall, the more open, the more expressed, but that's just an average.
So then why not just look at expression? Why is everybody looking at chromatin state? Why bother that? It all depends.
Well, I think most people are looking at transcriptome, at the gene expression, and actually the chromatin. The point is, the problem is that genetic variation, most of the genetic variants, they are not really within the gene, they are outside of the gene. They probably regulate gene expression, but we don't know why. We barely have any predictive models of why. And so a variant is probably opening something in your cells that doesn't open in my cells. And that's why some people have a disease and others don't. Yeah. Opening is, of course, part of this, right? It's the transcription factors that are binding. But yeah, one of the most scalable and cheapest ways of doing this is looking at the open chromatin. And yeah, it's the only way to really understand why a variant does this and the other variant does not do something. And so how people analyze this data typically is they define regions within our DNA that they call peaks. So here I define these regions, and then they're going to aggregate the data across this region. Then they're going to say, okay, this region is opening more, this region is also opening more, this as well, and they're going to use this as features. So these are called peaks. It's a high signal that stands out from the background. And then they're going to use these peaks to define, your classical feature by sample table. So you have like cells or samples here and then peaks over there. And then you have some counts and you can do whatever statistics you want on this. So you could, for example, use this data to then predict how it impacts the transcriptome or the cell state and vice versa, and also how the genetic variants are influencing this open chromatin. You can probably already see where I'm going to. Is this really the best way to do this, right? Because we know from deep learning, neural networks, we already know for a while that those models are probably often much better at defining features that are relevant, rather than as humans trying to kind of create these features ourselves. So can machine learning create better features? Nobody really tried that before, which was a bit surprising. Probably because, yeah, you need to know also a little bit about the biology before you can do this. And so we tried to do this. And first, so what did we try first? Well, we tried to look at the chromatin data to predict the transcriptome. So as a predictive model, quite similar as how you would use an image to classify the image. And so this is how our model looks like. So we start from the individual positions. So these are the fragments I showed before. For each cell, we have a bunch of positions. And then we use some positional encoding, which is very similar as what is used in transformers nowadays. So relative positions are transformed into some wider feature space that contains all the information necessary to link or to like focus on particular positions. This is then like put into a neural network as input features, which is then summed across for each cell, another neural network, and that we can use to predict gene expression. So, and these neural networks, what it will learn is quite similar as what we learned for the peaks, but now in an unbiased way. Namely, it will learn to focus on particular fragments that are over there, like right in the beginning of the gene, or maybe fragments that are in a very broad region. Like I showed in the example, it was of course a very simple example, this gene, this IL-1B gene. But for a lot of other genes, it's much more subtle. I will show this later.
So your gene expression is from pooled data then? Is that right? Is that why you're pooling all this information in the second neural network?
No. So our gene expression is also single cell. Yeah, so the profiling method we're using is a single nucleus, ATAC-seq and RNA-seq. We are pooling here across different fragments from the same cell and close to the same gene. Because sometimes maybe if you have a fragment there, it's not expressed. If you have a fragment there, it's not expressed. Maybe you need both to have the gene expressed. So we can also kind of learn combinatorial dependencies. So yeah. And so we found that this relatively simple model actually really outperforms all the rest. So compared to these peak-based approaches, which will aggregate across peaks, we often have a much better correlation with gene expression. Not for all genes, sometimes the peak-based approach is works very well, especially for the more obvious genes that have like clearly defined peaks. You can predict it easily, as a human could do it in Excel. But for all the other genes where there's more subtle things going on, which is actually the majority of the genes, we perform much better.
How did you split the training and testing data in this case?
I actually looked at different data sets. And so initially, like the training and validation is done within the same data set. But then we have test data sets from like the same system or another system, in fact. It's really tricky to actually transfer between systems. So for example, you have immune cells both in our blood and in the lymph nodes. But if you have the model on the blood cells, it doesn't really transfer well to the model on the lymph node cells, even though it's the same subtype of immune cells.
So the plot you showed in the previous slide is within a single cell type.
Across different cell types. Yeah. But so, I mean, you can use it if you have trained it on one blood data set. You could use it on another blood data set from a completely different donor. That works to some extent. And so we did this across a lot of data sets. And our method is actually outperforming the method that just counts the number of fragments close to the gene, which is actually the second best performing method. Kind of shows that using these peaks, you're not only removing information, you're actually doing worse than just counting the fragments, counting how much open it is. Now, one issue though with these models is we're using neural networks and we need to interpret, ideally. And so if you have peaks and you have a linear model, this is relatively easy to do. You just interpret, right? You have the peaks. But with a neural network, it's basically a black box. We don't really understand what these parameters are doing in there. So how we now interpret is, maybe it's not the best way to do it, but we removed certain fragments that had some characteristics, for example, from a certain region, we removed them. And then we checked how much did our prediction improve or worsen typically. And so one thing we can see, if we remove fragments that have a particular length, the predictive performance, like how much it worsens, actually really depends on the fragment length. And I mean, yeah, it is a little bit of biology, but bear with me. The fragments, they basically gonna span these nucleosomes, these structures that are in the DNA that kind of wrap it. And that actually, these structures, they close the DNA. And so if you have a certain fragment length that is about the length of the DNA that is wrapped around this structure, it actually means that this is closed. And so often if you have such a length, which is about 180 base pairs, your model is actually less predictive for gene expression because it means there's something close there. So, yeah. Now, we also, for interpretation, we also kind of did a scanning window approach where we removed fragments when we scanned through like close to a gene. And then we looked at what could you see, which regions are actually important. And so here you kind of see a distribution of the fragments. So most of the fragments are located here. And then here you can also see the peaks that are used. So like it's really focusing on peaks. But then here you can see how the importance of a particular region for predicting gene expression. And what our model actually picked up is that, okay, this region is very important. It's also a peak. But then there is this other region, which is right before this region that was actually, it had a lot of fragments. That's also very predictive. It's right before this. It's sometimes within a peak, sometimes outside of a peak. But it really does show that these peak-based methods, they just fail to capture a lot of important biology. Because this region then, if you actually look, there are some genetic variants over there that are actually implicated in disease. And if you would have used peak approach, you would just have not seen it ever. And then if we then look at the raw data, I always like to look at the raw data, right? You can indeed see that. So here's again the gene expression. Gene is not expressed. Once the gene is expressed, this region is actually open, right next to the region that was already open. Okay, so the conclusion of this part, where we try to predict the transcriptome from a chromatin state, is that even though we have used a simple neural network model, we can already outperform the state of the art. Classical methods, they remove information. That's kind of the classical story where you do your own feature selection. It removes information. Humans are indeed not so good at feature extraction. We think we see these peaks, but in reality, they're not always that relevant. And then interpretation can be a bit more difficult, but not impossible, right? You need to use, for example, some perturbational things, but you need to do it after you have created the model. You cannot do it a priori anymore. But I think that's a fair trade-off.
Could you have some kind of like weak inductive bias in the model that you start with? It allows it to be more interpretable.
Yeah, perhaps.
And that's what a lot of people are doing now, right? Trying to include components of the model that are more prior knowledge about system study.
Yeah, for sure. I think these fragment lengths, we could include that. That would be the obvious choice. Yeah. And then it would improve even more. Or it would improve interpretability. Yeah. No, that's certainly fair. Yeah, people, yeah.
Would we have considered using functional regression for this? And that would be very simple, and that would capture the interactions.
Yeah.
And that would be a very simple way of
a good baseline, right, compared to
peaks. I mean, it would be much better than peaks, and it would be much more interpretable than neural networks.
Okay. Yeah, I don't know. I'm gonna look it up. I don't even know functional regression. Like I haven't heard of it. The only thing that people have tried to do is using a scanning window.
It's linear regression, but like in the continuum, basically.
I see. Okay.
You would have to use the appropriate regularization. You basically learn functions. I'd be happy to talk.
Yeah, sure. Is it enclosed to Gaussian processes or not really? No. Yeah.
Simpler than that.
Yeah, simpler. Okay. So then for the next part, we're gonna look in the other direction. So here we're trying to learn how the chromatin state changes with the cellular state. So what's the cellular state actually, right? Oh yeah, it comes later, but a cellular state is, for example, the type of cell. Now, if it's a skin cell, neuron, immune cell, everything. How does then the chromatin state depend on it? And so this is again the classical way that people have approached it so far. So they used peaks. And then they try to look across different cell types. So these are different immune cells in our blood. They try to look how these peaks change. Yeah, so if it's here red, it means the peak went up. If it's blue, the peak went down. It's basic linear regression. Now, visually, you can already see that you're losing a lot of information. Like most of the fragments, or half of the fragments, let's say, are not captured within a peak. And then even within peaks, you see some heterogeneity. So we try to improve this. And look, try to improve, or just look whether we can model it, and then if it actually improves our biological interpretation. Again, can machine learning create better features? So the way we approach this is you can consider this, of course, as a distribution, a one-dimensional distribution that changes between the cell types. A histogram that I showed in gray in the last slide is also a distribution. But this distribution, it can have like various forms, right? Sometimes a very broad region can be open, sometimes a very narrow region can be open, and this region can change between different cell types. So the resolution of this changes between different cell types. And so we found like one way to model it is, for example, using mixture models, but another way could be to use normalizing flows. One-dimensional normalizing flows, which are effectively, these are transformations of very simple distributions. So you start from a simple distribution, for example, a normal, but in our case, it's a uniform. And then you transform this distribution in several steps until you get a complex distribution that can have peaks and valleys. And the way you do this transformation is very important. You cannot just use any function here. You cannot, for example, use a neural network. You need to use a bijective function. For example, a monotonic spline. That's a prerequisite to actually be able to transform probabilities. And here I want to kind of digress maybe a little bit. I think prediction is cool, but being able to estimate some density is even cooler. I honestly believe that this might be one of the futures of machine learning, is to actually be able to estimate what is likely instead of just being able to predict one thing. Anyway. And so how does our model look like? So we model the distribution of the chromatin accessibility in a particular cell state through multiple layers of monotonic quadratic splines. We transform a uniform distribution Z into a complex distribution. And the key thing is that we do not assume a priori a resolution. So we have several layers of transformations, and kind of the model can decide which layer it tries to focus on. And each layer has different resolutions. So for example, if you have 32 knots, you're looking at broad regions. If you have 128 knots in your spline, you're looking at very narrow regions. And the parameters of these transformations, they can change depending on the cell state. Yeah. And to actually add some statistics in here, because that's always the tricky part, this model can easily overfit. If you have like a couple of cells, it will just find whatever. In some specific regions, it will find a very high likelihood. So what we do is we put like a Bayesian prior on this, on the parameters here, which is then like the parameters of this prior are estimated using some partial pooling to ensure that it's kind of a regularization. Okay, and so as I said, this can have multiple resolutions, and it can decide the resolution wherever it's needed. And I really like this model because it's quite similar as neural network. You use multiple layers, but instead of using multiple nonlinear layers to predict something, you're actually using multiple nonlinear layers to estimate a probability. But for the rest, you can kind of intuitively think about it in the same way. And so this is how various models do. And so these are all peak-based models. This is like a rolling window model. It works, a sliding window model. It works fine, except in some cases. But then this is our model. And it's able to like find broad regions and also narrow regions of chain. And then given that we're biologists, we try to then understand, does this actually perform better? Maybe we're just seeing useless things. So we evaluated this using various approaches. For example, we looked at genetic variants. Are these then more enriched within the regions that change in our case? Yes, about 1.5 times more. Are also functional binding sites more enriched within our regions? Yes, and it's always about 1.5 times better performance, which is quite a bit compared to the state of the art. And of course, we also want to understand why then does our model predict better? Like, what are then the assumptions that are being made by these other peak-based methods? So it turns out there are mainly two things. So the peak-based models, like this is an example where here there was a peak, and we as humans defined, look, there's a peak over there. It's not changing. It's actually the region right next to it that is changing. But the peak thought, oh, this is just background. It's not really relevant. But it turns out that can be very relevant. There are a lot of binding sites right in this region. At the same time, imagine that actually the peak method looks like this. Then it will see, yeah, this region is changing. This is a differential peak, so to speak. But a lot of, like half of the peak is actually useless, or not necessarily useless, but it's not changing. And so there are also a lot less functional binding sites in those regions. So these are like two main assumptions that are made by these methods. And because of those assumptions, our method performs better. Now I'm going to look into the mountains in our DNA. Because how do these regions actually look like? Because if you then look at the data, you can see various different topographies, let's say, or shapes. And in this case, I kind of define them myself manually, but maybe in the future, we can actually learn some predictive models that use these shapes, or just, some convolutional neural networks, for example, that use these shapes to predict something else.
How much of these shapes are noise versus really well-defined contours that are meaningful? I don't know how you come up with these peaks in the original data set. But can you comment on that? And we expect it to be extremely precise in terms of regulation of DNA access.
Yeah, that's a good point. Wow. In any case, like we're looking at this statistically. So in principle, if there is noise, it shouldn't capture it. Like we can test this using a validation data set that indeed, then suddenly in the validation data set, the predictive performance is equal. Whether it's functional, that's another question, of course, right? Some regions might just open because there's a binding site there, but it doesn't really change anything. There's a gene regulation. That's exactly what I wanted to look at here as well. Maybe some types of regions are more noisy, and that we could then relate to their shape. So yeah, these are like two of the basic shapes. Let's say they are the peaks and the chains, and they are like your typical peaks that are opening. So here in red, you see the change that is happening in a particular cell type compared to the baseline. So like the average accessibility across all cell types. You can see broad regions that are opening with multiple peaks or narrow regions that are opening. And we did some functional validation by referencing other data sets. We found that they are regulating the DNA, but they are actually not that important. They are not the most important. Even though the classical methods really focus on these, funnily enough. because we thought as humans that these were important. You have other types of regions, like for example, we call them the volcano. It's just a name we give to these things, but they are de novo opening. Yeah, so the DNA before wasn't really open or was barely open, but now it's suddenly opening. And these regions are really truly functional because it's bam, suddenly it's open. Before it wasn't. And we found really strong enrichment for whatever marks that are important for like activating gene expression. Then we have a couple of others. We have, for example, the flanks, and we have the ridges. Some of these have functions. Some of these are a little bit, we don't know. I think if I would have to bet which ones would be noise, maybe the flanks are noisy. But it's just a bet. We don't really know. We would actually need to mutate these regions to be able to understand this. For now, it's just a bit of circumstantial evidence, really.
To mutate them, or can you just like virtually mutate them in the sense of, like, for example, if you use attentional networks, you can see which regions that they actually attend to. You know what I mean? So you can do in silico measurements to see if they're likely to be important or not.
Yes, but do I trust those networks?
And they're predictions, right?
Yeah. Yeah, so we could certainly. That's an interesting thing. There are these models that use the gene, like the genetic code itself, the DNA sequence itself to predict gene expression. Still, I mean, it's based on not that many samples often. So for individual mutations, I'm not sure whether I trust them much. Although it could be some extra evidence, that's true.
Yeah. Missing something. So there's two parts to where you talked about. The first part was about creating a model that can make good predictions, transcription. And the second part was more about fitting this complex landscape of chromatin accessibility with normalizing flows.
Yeah.
But what I'm wondering is, these are two kind of separate things, but the first part can be applied to the second part.
Ah, that's fair. Yeah.
For which regions of this landscape are actually critical.
Yeah, that's fair. Yeah, we can do that. We would still need to do that, indeed. Yeah. Although, I mean, it would still be something correlative, right? I mean, even the first model is not truly predictive. It's just correlation. Yeah, but it's completely fair. If you would want to know whether this is truly a causal region, yeah, we would need to kind of mutate, I think. Yeah. There are also a couple of other regions which actually, like, they're a bit weird. Sometimes you have, for example, a piece of DNA that is opening close to another DNA that is opening. These are not well enriched, but these are super predictive for gene expression. I'm not sure what is happening there. We kind of think that given that this peak is close to another, it kind of amplifies a signal close by. Now you need to think about this in thermodynamics. Maybe given that something else is opening, this one is kind of amplifying the signal.
I mean, just a naive question. I mean, I'm not sure I understand exactly what your visualization means with the different colors. You have two curves on top of each other. Maybe they cross. So, I mean, from your explanation, I sort of understood the fact that you are, learning a model of distribution which would correspond to a single curve. So, can you clarify maybe what are the colors?
Yeah.
What are the two curves? Sorry, maybe I.
No, it's a good question. Thank you. So in the dotted curve, that's the baseline accessibility. That's the baseline distribution. It's kind of the mean of all cell types. Yeah. And then the red or the blue is how much this changes in the cell type compared to the mean. So the red is, for example, in monocytes, in this gene, this region opened all of a sudden. While on average, it was like this. It wasn't really that open. And then the blue is that it closes. So it's the opposite direction. So normally, I mean, we have actually multiple of these right on top of each other across different cell types. And if something is opening in one cell type, it's closing in the other. All right, and so we found these other topographies as well, let's say. And we could kind of relate them to multiple functions. It's actually kind of cool. Like, for example, these canyons, which means that something is opening, but close by something is closing. it's really strongly related to repression, apparently. You might think that like this region might be important and to activate the gene, and the fact that this is opening is probably closing the nearby region, but it's really hard to interpret still. A lot of this is unknown. We don't really know exactly how this works, especially at this narrow resolution. But anyway, to kind of conclude, we found here as well that classical methods, that they remove information, and that even a one-dimensional normalizing flow can capture the full complexity, as far as we know, of differences in accessibility between cell state, and that the shape really matters. Classical methods, they just assumed it's a peak, but in our case, we found different shapes and they matter for different functions. And what I really love about this is that machine learning, it not only can be used to just predict, but it actually enables new discovery. We didn't really imagine that you had such a variety of shapes. Okay, and that brings me to, how far am I? Right. To the last part, which is to predict which is, can we actually now use the genotype to include the genotype in this as well? So genotype is our DNA sequence. Can we now add this new information about the chromatin state to better predict the transcriptome? And I mean, why do we want to do this? Well, as I showed before, genetics is really important in diseases. But the problem is, even though we know it's 50%, we don't really know which 50%, which mutations is it exactly. And in particular, when are these mutations active? Which cell types, when, and how do they work? We can maybe answer this for about 8% of mutations. So a lot of studies have been done here. Billions of dollars have been spent on this. We maybe know it's for 8%. What about the other 90%? We don't know. And so, I mean, the thing I already saw, I try to now correlate these different shapes with where these functional mutations are. So there are some ways to experimentally test which mutations are functional. So you can use this. However, these experiments are really expensive. You need to basically sample 1,000 patients. Well, I already did see that some shapes are more functional than others. And so I'm kind of thinking about creating now a model that actually uses shape information to predict which mutations might be more functional than others. And this would really be cool, because then you could look into a patient and actually get data from this patient and then try to test or try to predict at least which mutations might be more functional than others. Okay, so I also had another part, but maybe it's best to leave that for another time. I still have time? Oh yeah, okay. Well, it's actually a completely different project, but it's related in the sense that now we're looking at the transcriptome and the cell state yet again. But it's more a project about how we discover things within our data, within our very highly multidimensional data. And here we try to investigate the cell state. So a cell state can be various things. It can be like a cell type, like I just described. But it can also be a position of the cell type, but often it's kind of continuous phenotypes. So for example, a cell can be differentiating, a cell can be cycling, or it can be stress, whatever. And the key thing is that the cell does these multiple things at the same time. It is a particular position, it's a particular type, but it's differentiating while it's cycling. And so you can see this is a highly dimensional space. And we want to understand this because this space is often really perturbed in diseases. This is an example of an experiment we did in the lab where we overexpressed. So we kind of activated a particular gene, PAX9. It's a tumor suppressor gene. It's called a transcription factor, so TF. And so here in gray, you can kind of see the cells that did not get this gene. And then in the colored space, you can see cells that were given this gene. So this is a two-dimensional representation of how the cell state changes in a human. Then you can see that, okay, this is the cell cycle. Here you have cells that are proliferating. Here you have cells that are not proliferating. They are set apart. That makes sense because proliferation really induces a different cell state. Then if you look at the expression of another gene, CBP beta, which is an important gene that is related to this PAX9, you can see that it's only actually upregulated in these cells and not really in these cells. So in the cells that are proliferating, the gene is not changing anything. So it means that there is kind of an interaction between PAX9, which we induced, and the cell cycle. So two different cellular processes or two different cell states, the PAX9 and the cell cycle, they're interacting molecularly. Okay, so that's just an example of how these interactions happen. And these interactions are really important in cancer because, for example, PAX9 is a tumor suppressor gene. It's known to reduce proliferation of the cells. But how does this exactly work? We don't know. And so more generally, we can make different models of how this interaction between the cell cycle, which is shown here, and the transcription factor, which was, for example, PAX9, works. In one case, it could be that the cell cycle inhibits the transcription factor. In another case, it could be that the transcription factor inhibits in some way the genes that are upregulated by the cell cycle. And these two hypotheses, let's say, could be represented like this in 2D. So these are different types of interactions that we actually observed in our data. Where here this gene is downregulated during the cell cycle, even though it's upregulated by the transcription factor. Well, here you have a gene that is upregulated in the cell cycle, but only only when the transcription factor is there. And the problem is that we're dealing with is that these are just two examples, but there are really a lot of examples. And the thing is, a priori, we don't even know what we would expect to find. These are things we found in our data because we looked a lot at it. It's hard to kind of define beforehand what might our hypothesis be. Still, we want to kind of delineate what are the different patterns we see. And I mean, the tricky thing is, like, initially you could try to build linear models here. So it's a classical, you have some interaction between multiple few features. But the thing is, it doesn't really fit well within our paradigm because here you need to define your hypothesis beforehand. And also, a lot of these hypotheses are not really nested within each other. Like, for some genes, the cell cycle might do nothing, while for others, the interaction work in this direction. So we cannot use very well this linear model, classical frequentist testing paradigm. We think we need to kind of go more Bayesian to solve it. So we looked into this Bayesian workflow, which is kind of a way to test models iteratively to learn more about your system. So what you start with is you define multiple models. You need some models to begin with, but these can be very simple ones, like there is no interaction. Then you train it on your data, which means you learn the posterior. Then you compare these models, but now in various ways. So first you do your typical statistical check. So you make sure that if you would redo the experiment, you would still get kind of the same result. You also do your biological check. Is your model, like if you have two models that are a bit different, for example, there's an interaction, there's no interaction, but is this actually substantially different biological, or maybe just about the same thing? And then finally, important, you also do a predictive check, which means that does our model actually fit the data well? Maybe it fits much better, but still it's a very bad model. So it doesn't really mean it's capturing the biology. Then you adapt your models based on this. And so the first two things are relatively easy to do in the Bayesian paradigm. Effectively, your statistics, you can do that by, for example, comparing your marginal likelihoods. your biological check, well, you can check the effect sizes, right? And you can define some cutoffs, for example, based on that. The predictive check was a little bit more difficult because classically this is actually done by looking at the distribution of your data. Like, what does my model predict? And you look at the distribution of these predictions. You check, does this correspond? In our case, our models are really like, they are multidimensional predictions, and they are count data, for example, which is not easily visualized. So that didn't really work well for our data. So what we went for is, yeah, we don't know actually whether this is the best approach, but still, it did work in our case, is we actually defined some black box model based on a neural network, Bayesian neural network, that tries to model this transcriptome based on all the input variables. And then we compare both. So we compare our models our hypothesis that we predefined and our black box model. And if the Bayesian neural network fits the model much better, the data much better, that means that, yeah, probably our models are not very good. And we need to add some extra models. We need to look at what's happening to our interpretable models and maybe define a new one. So yeah, that's our final workflow. And it does work. I mean, in this case, it did really work well, because in the sense that the different models that I defined before, which was like here, initially, we just defined 2 of them. But in the end, we had to add multiple extra ones based on what we saw, because our model wasn't really fitting well to the data. Okay. And so we have a couple of other use cases here as well, where we, for example, we looked in the liver, how the specialization of cell types is different between multiple diets. Again, a lot of models, how this can change depending on the diet. And we use this to kind of find out that it's only after a couple of weeks that we actually see a substantial loss of spatial specialization in the liver. While in 24 weeks, it's mostly just adaptation. Now, the key thing is, though, that like, why do we actually need these models? Well, the problem we're dealing with is that we have a lot of data. So this is the data set that we actually used. I showed you now PAX9, which is somewhere, I don't even know where it is here, but we have data from hundreds of other transcription factors as well. And each one of these can have different models. And so it's really hard to automatically or to analyze this manually. You cannot. So you need to use some kind of more automated approach that you can test multiple models and see where, in which transcription factors these models fail. Right, and so our dream at some point is to kind of create this Bayesian toolbox that you can use, not like multiple cellular state variables, but also multiple things to predict. Like, we now looked at the transcriptome, but we want to look in the future at multiple modalities as well. So that we can kind of have this knowledge discovery toolbox that can be used across all of single cell biology. All right, to conclude, there's still a lot to discover about the link between different cell state variables. And this is just one example of the differentiation and the cell cycle. But we think the Bayesian workflow is really suited for this. And there are, however, still several open challenges regarding the implementation, because it's not easy to create such a tool. With that, I want to thank you, and I want also to thank my colleagues for all the help and for their ideas. And thank you.
Thank you very much for your talk, Wouter. Next opportunity is actually on campus. In 2 days' time, we have the EPFL Industry Day, the very first since 2019, as far as I know. There are many great speakers lined up, quite some actually affiliated to CI, so please feel free to pop by. There's also a roundtable on AI for engineering in the afternoon, which we're happy to host with colleagues from several faculties involved. Then on Thursday and Friday this week, so it's the 9th and 10th March, we are looking forward to actually welcome the colleagues from Recon AIP in Japan. So this is going to be 2 days intensive workshop, more on the applications of machine learning. And then on the 13th of March, we are looking forward to our CS and drug seminar with Professor Michelin, who is affiliated to CHUV and to UG, so the 2 cantonal hospitals in the canton of Vaud and Geneva. Yeah. Thank you. I look forward to seeing you soon again. Goodbye. Goodbye.